Okay, for the second half, I'm very happy to introduce Ilan Schwartz, who currently is finishing up his PhD work. He received his MD from the Queen's University in Canada and did his ID fellowship at the University of Manitoba. Did a lot of work in Cape Town and has become an expert on Emmonsiosis, which is a new pathogen to many of us and very similar to many aspects of histoplasmosis, and also has some interesting data on Blastomycosis, which they see a lot of in Manitoba. And so, Ilan, welcome.
Okay, so first of all, thank you so much for the honor of being able to talk to you today. And this really brings me great pleasure to talk about some work that I think is really exciting and really should make us question what we know about the endemic mycoses and about their causes. So the story is essentially one about disease. Cases occasionally were reported from other domains like Africa, India, the Middle East, but it was uncertain what to do with these cases, what to make of them. Were they identified correctly? Were they truly autochthonous, etc.? So eventually these cases accumulated, and it caused some important questions to be asked about whether really what we're looking at is one disease or two, specifically asked in the context of this Lancet editorial with regards to blastomyces in North America and in Africa. And essentially, are we really comparing apples to apples? And what I'm here to try to convince you today is that blastomycosis is not one disease and it's not two diseases. It's actually many diseases. So our knowledge about the genetic diversity of blastomyces has really advanced in recent years. So building on the work of previous investigators, Jennifer Meese from Marshfield, Wisconsin, performed multi-locus microsatellite sequencing on 112 clinical, veterinary, and environmental isolates, mostly from the Wisconsin area, generally from North America, and occasionally from elsewhere. And their findings are here. Essentially, they demonstrated that there were two very distinct genetic groups. Another group, Susan Richardson and colleagues from Toronto, they also analyzed large groups of isolates, and they used multi-locus sequencing. And in addition, although they just had 78 isolates, they also included some from domains further afield. They confirmed the findings of Meese and colleagues in that there were two distinct genetic groups. So I've put them in different colors here. So this is one group, this is one group. But they took their analysis a little bit further, and they invoked a concept called genealogical concordance phylogenetic species recognition. And basically this concept is that when you have the gene trees of multiple different, when you have multiple gene trees that are overlaid and compared, the transition of the topology from congruence to incongruity defines the limit of species. Now, they sequenced 5 loci, and 4 of those demonstrated that incongruence, and that's how they determined that there was the existence of a cryptic species, which they named Blastomyces gilchristi. So, this was raised yesterday many times. Who cares? And it's a very valid question, especially for clinicians. So as far as we know, these two species are morphologically and physiologically identical. Are there clinical differences? Well, Jennifer Meese's group tried to persuade in a JID paper that there were, but it was uncompelling, didn't withstand multivariate analysis. So none that have emerged yet. But there is definitely some epidemiological significance. So specifically, the areas of the world that have the highest proportion of isolates that typed as Blastomyces gilchristi are also the areas that have the highest incidences of blastomyces. So specifically, Eagle River, Vilas County, Wisconsin, and Kenora, Northwest Ontario, both of which have incidence rates of more than 100 cases per 100,000. So what about Africa? We know there's at least 19 countries in Africa where blastomycosis has been diagnosed, in addition to Israel, Saudi Arabia, and India. And there's important differences that have been noticed over the years by various investigators. So one is that there seems to be different morphology. The conidiophores of the African strains seem to be shorter, and they have conidia that cluster on the end of them in florets. The conversion from the mold phase to the yeast phase seems to be much more difficult with African strains in the literature. And in early cross-mating experiments, McDonough and colleagues demonstrated that there was failure of true sexual spore formation when attempting to cross a North American and African strain. And important work by Kaufman and colleagues demonstrated that there's important antigenetic differences. So in North America, 88 out of 88 isolates that were tested expressed the A exoantigen, whereas in Africa, only 1 out of 12 demonstrated the A exoantigen. 2 decades ago, Geho and colleagues demonstrated important genetic differences using DNA melting curve analysis. So the top curve here is the North American strain. The bottom curve, which is a completely different shape, sinusoidal, is an African strain. And then there's a very fine dotted line running alongside that, which is the curve for Emmonsiosis and Paracoccidioides. Clinical differences have been reported, most notably that there's a tendency of African disease to localize to bone, and according to some reports, it's the most common reason for presentation. And in stark contradistinction to the case with blastomycosis in North America, there's actually never been a reported case of blastomycosis in an African animal. In Africa. Because if you take an African animal and you move them to Tennessee, like a cheetah and a lion in this one study, they can and do get blastomycosis. So what does the typing tell us from these previous studies that I mentioned? Well, the MLST analysis tested 3 isolates, and 2 of them were placed within Gilchriste and 1 within Blastomyces dermatitidis. However, I just want to stress that this is obviously a very small sample size, and there were additional isolates that didn't fit within the expected findings that weren't included in the further analysis. So in order to address this question, led by Nilesh Govender at the NICD in South Africa and his doctoral student Sidiso Mapanga, we went and looked at old isolates of Blastomyces in their stores from the last basically 45 years. And we've submitted those isolates to MLST. We reviewed cases where available, and we've got the whole genome sequencing of one of these. I'm just going to walk through 2 of the cases with you just so that we are on the same wavelength. So the first case is an HIV negative individual from Cape Town in the Western Cape. So he presented with a 4-month history of cough, hemoptysis, and weight loss that failed to respond to empiric TB therapy. On examination, he had fleshy skin lesions on his face, and his chest x-ray demonstrated a cavitary lesion in the right upper lobe. He had a bronchoscopy which demonstrated endobronchial mass in the right upper lobe bronchus. And that was biopsied. And this is what was seen on the histopathology. So there's the typical broad-based budding yeasts with double refractal walls. And this was called by the histopathologist as blastomycosis. Now the isolate that grew in culture was identified phenotypically as Blastomyces dermatitidis. The second case is also an HIV uninfected gentleman, also from the Western Cape in the winelands. He worked at a wine factory. And interestingly, his dog died just before he became sick. So could that have been the first case in Africa reported? I don't know. He presented with a 7-month history of gradually worsening headaches associated with some intracranial hypertension. And he had a 2-month history also of ataxic gait. So his chest x-ray showed bilateral pulmonary infiltrates with perihilar shadowing. And the MRI of his brain demonstrated a solid cerebellar mass with microabscesses. So those were biopsied, and the histopathology again showed these broad budding base, these yeast with broad base budding and thick double refractile walls. And this is the mold that was grown. So phenotypically, it was also identified as Blastomyces dermatitidis. So just want to make sure that we're all on the same page and that we all have the same diagnosis here. I think the clinicians and the mycologists, the microbiologists will agree that this is Blastomycosis. But when we went back and looked at the morphology again, the conidia actually looked a bit different. They appeared in clusters, in florets. And this is a lot more consistent with an Emmonsia species. This is actually the light microscopy image. And just to give you a reminder of what Blastomyces dermatitidis is supposed to look like with the conidias and the conidiophores. In addition, it was difficult to convert to a yeast phase in vitro, and it lacked the A antigen. So we sequenced the ITS region of the ribosomal RNA gene, and the identification came back as Emmonsia parva. So right now you're thinking, okay, wait a minute. So you're telling me that African Blastomyces DNA melting curves are more similar to Emmonsia than to North American Blastomyces. The morphology here is more suggestive of Emmonsia. The sequencing is Emmonsia. This is not a Blastomyces, it's Emmonsia. But the thing is, they're the same thing, particularly Emmonsia parva. Emmonsia parva is a sibling species of Blastomyces. The only reason they're not in the same genus is because Blastomyces as a genus name is invalid. So if they were to join together, they would become Emmonsia. And you guys wouldn't be happy with that. So this is the results of the whole genome sequencing, courtesy of Jose Munoz and Cristina Cumo, of the brain strain. So you can see that it does cluster here beside Emmonsia parva and Blastomyces dermatitidis / Gilchrist. We also found that it has it's nearly this exact same as an isolate that was recovered in 1993 from the mucocutaneous lesion of a man from Israel. And this, we'll call here species number 3. Now, Joe talked yesterday about the dramatic genome expansion that was seen in Blastomyces, and Jose in Christina's lab has confirmed that, in fact, that's not seen in this organism. So this organism has a contracted genome or a normal-sized genome, the same as histoplasma, Emmonsia parva. So for some reason, it hasn't gone through that genome expansion. So I'm just going to take you back now to North America. So in 1970, a case of Blastomycosis was diagnosed in Alberta, a region of the country, a region of Canada, not known to be endemic for the disease. And it was a middle-aged farmer with a history of alcoholism and possibly diabetes who presented with what turned out to be a fatal encephalitis. So the post-mortem tissue from the brain and lung both demonstrated the presence of these yeasts with broad-based budding. But there was something peculiar about the organism that was isolated. The authors, Seekin and colleagues, noted that it wasn't typical for Blastomyces dermatitidis because it rarely produced any conidia. It formed spirals of the mycelia. It was difficult to convert from a mold into a yeast. And in animal experiments that they performed, it seemed to be of low virulence in mice and guinea pigs. And in fact, this is also a new species. So in addition, this has since been isolated in people from Saskatchewan, Montana, California, Colorado, Texas. So again, are we talking apples to apples when we talk about different blastomycosis from different parts of the world? Well, no, because blastomycosis is actually more of a fruit salad. Now, let's just go back and talk about that peculiar organism, Emmonsia parva, for a second. So I'm going to talk about the genus Emmonsia, which until recently contained these two very odd organisms, Emmonsia parva and Emmonsia crescens. Now, they're very similar to one another. Both of them are dimorphic. In the environment, they exist as a mold phase, presumably in the soil. When the conidia are inhaled, usually by small animals and rodents, but very occasionally humans, they don't transform into yeasts. Instead, they undergo a dramatic expansion in size and they form these large resting cells called adiospores. Now, adiospore, by definition from Latin, means they do not disseminate. They do not sow by seeding. Occasionally, humans can become infected by inhaling, and this is a disease called Mycoses, but it's exceptionally rare. There's only 50 cases or so in the literature, but it's very common in animals around the world. Now, both of these organisms cause this disease. They're really only distinguished on a few different phenotypic factors. One is the tolerance to cyclohexamide, one is the size of the yeast. In particular, Blastomyces crescens can grow up to, the adiospores can enlarge up to 500 microns, which is a million-fold expansion, and by the temperature at which they convert. But what you need to know, and why this is such a confusing issue, is that these organisms, in spite of the striking morphological, ecological, pathophysiological similarities, they're actually not very closely related at all. So for a long time there was debate about whether they represented one species with two varietals or two different species, but now we know they don't even belong in the same genus. Things got more confusing in 1998 with the diagnosis of disseminated mycosis in a woman from Italy with advanced HIV disease. Now, in tissue there were small yeasts in skin lesions, and the organism that was isolated, it also, the conidia came on little florets. It morphologically and genetically was clustering closest to Emmonsia crescens. So this was determined to be a new species of Emmonsia, and it's been called Emmonsia pasteuriana after the Pasteur Institute. So now we've got these two different disease syndromes caused by this one genus of poorly related organisms. And now this is a true thermally dimorphic organism which is capable of extrapulmonary dissemination and small yeast forms. But there was only ever this one case in Italy until recently. So in Cape Town in 2008, a young man was reviewed because of the development of widespread cutaneous lesions that started soon after initiation of antiretroviral therapy. A skin biopsy was performed and innumerable small yeasts were seen, and really the absence of an inflammatory response. The organism grew as a mold. You can see over here its morphology. And the ITS of the rRNA was sequenced. And again, what came back was a diagnosis or an identification of Emmonsia. And this was determined to be a new species that clustered most closely to Emmonsia pasteuriana. Now, the first case was followed by several others that trickled in, and the first 13 were reported in 2013. So this is about when I came on the scene, and so at first we wanted to understand the epidemiology of the disease, better document the clinical histories, and try to understand the ecoepidemiology. Where does this organism live in nature? So I performed a retrospective chart review of 54 cases that came from across South Africa. You can see the vast majority came from the Western Cape, but there was certainly a referral bias. And now we think that likely the organism and the disease occurs throughout the country. All patients were profoundly immunocompromised. There was one transplant recipient, and the remainder were HIV infected with a median CD4 of 16. Skin lesions were present in 96% of patients where this was ascertained, usually very dramatic, although they could be less obvious, and presumably there's many more cases that are missed. Lower respiratory tract infection was surmised based on abnormal chest x-ray in nearly 90% of individuals. The diagnosis was best made with skin biopsy for histopathological examination. There was nothing pathognomonic on the staining, but at least to have these patients recognized as a fungal infection, so we could stop their TB therapy and get them on the right therapy. In addition, these samples were the best, had the best yield for culture, although we did manage to isolate it from fungal blood cultures in a minority of cases, in aerobic blood cultures and even fewer. I can confirm that there was some cross-reactivity with the immunohistoplasma urinary antigen kit. Okay, so as I mentioned, most patients were presumed to have tuberculosis based on their symptomatology, and that's reasonable given the epidemiology in the region. So 75% of patients were on TB treatment, and about a quarter of patients were not diagnosed with a deep fungal infection, at least not until after they died, when their blood cultures came back as positive. In total, half of all patients died in our series. The patients that received amphotericin B followed by itraconazole seemed to do, reasonably, but obviously this wasn't a trial. So next we turned our attention to try to identify where this organism was in the environment. So we've studied the soil and the air for evidence of the organism, and we've studied rodents and shrews for evidence of naturally acquired infection. I'm just going to present the soil findings to you. So we collected samples using a convenience sampling from around the country, but mostly from the Western Cape. And we submitted those both to culture and to molecular detection using a nested PCR with ITS followed by species-specific primers. And we collected 60 samples, and you can see here the green dots indicate that Emmonsia nucleic acids were detected. The red indicates they weren't. And in total, 18 out of the 60, or about 30%, it contained Emmonsia. Most of these were from the Western Cape, and there was about a third of all soil samples. And in this map, the purple dots are additionally the overlay of where the cases lived. Sorry, I don't know why that didn't turn out right, but all soil samples that were tested did have evidence of the organism. Now, we also tried to culture the organism, and we used both direct plating and flotation method, and unfortunately those were unsuccessful due to rapid contamination by rapidly growing bacteria. However, we have now developed a mouse passage model to passage that soil through mice, and it actually performed very well in pilot studies where we've now tested 20 samples, and those results are pending. So I just want to stop here just to reflect on the landscape of what's happening with these organisms. So over the last couple of years, so as I mentioned, there were 54 cases in that initial series. We've now diagnosed about 80 cases, and there's many more that have gone undiagnosed, all in South Africa. In addition, Emmonsia pasteuriana has been identified in Spain, India, and China. Another species has been identified in China. Another novel species has been identified in Germany. These are just one case each, but phylogenetically distinct. And another couple cases of a new species have been identified in Saskatchewan in Canada. So when you add to this the Blastomyces-like organisms, which have cropped up in South Africa, Israel, and in Canada and the United States, we're talking about a large swaths of the globe that are affected by newly emerging organisms. So with all of these novel species in the Mycoses-Ilan, there's going to be impending taxonomic shifts. And I'm just going to walk you through what I anticipate is going to happen. So the genus Blastomyces is hopefully going to be conserved. So hopefully we'll still have that as a genus name. But there's going to be at least 2 new organisms, the ones that I've told you about. And in addition, Emmonsiosis parva will be reallocated to Blastomyces. Now, one issue with that is Emmonsiosis parva is actually the type species of Emmonsia. And because of naming rules, when the type species goes, actually the name goes with it. So it's taking its name with to Blastomyces. Emmonsia is going to be a synonym. And now everything else that's been called Emmonsia in the meantime needs a new name. So all of these organisms that cluster near Emmonsia pasteuriana, the ones with the small yeast and cause disseminated disease in immune-compromised hosts, are going to be given a new name. The proposed name by Sabrin is Emergomyces. I don't know if that'll stick. And the type species is going to be pasteuriana. And then finally, the fate of Emmonsia crescens is really up in the air. Lynn Sigler confided to me the other day that she lies awake at night worrying about this organism. It's going to be so lonely. So takeaway points. So there's 2 groups of novel, medically important dimorphic fungi in the Mycoses-Ilan. One of these groups is related to Blastomyces and causes Blastomycosis. One of these groups is related to Emmonsia pasteuriana and it causes a disseminated disease clinically identical to disseminated histoplasmosis. And again, getting back to the discussion of, well, why does this matter? But it's my belief that really only once we disentangle these organisms from one another can we truly understand them and learn how to best prevent disease and diagnose and care for infections in our patients. So with that, I just want to thank all of my collaborators, of whom there were many, and who have agreed to me presenting a lot of unpublished data. And I want to thank you again for your attention.